Welcome to the American Cannabis. I'm your host, Ellis Smith. I'd like to thank our sponsor, Cannabis Tech. Today's topic is going to be Cannabis Germplasm 101. I've got a great group of folks on a panel today. Dr. Mitch Day will be really managing the panel today and being the host for this. We've got Dylan Wilkes, we've got Max Jones, and obviously Dr. Mitch Day. We're going to have a little different format today where I'm going to have Dr. Mitch Day kind of run the panel, and I'll let him give everybody a chance to introduce themselves. And excited to really get into this topic as this is really a big conversation right now in our industry. And so thank you everybody for getting on. And Mitch, I'm going to pass it over to you and we'll kind of start the panel. So thank you everybody for coming.
Well, good morning. Thanks for the chance, Ellis. I always appreciate working with you. Thanks also to our panelists. We have Dylan Wilkes. He's the founder and CTO of Orange Photonics, makers of the Light Lab. I'll let Dylan introduce himself. And then we also have Max Jones. He's an assistant professor at the University of Guelph, an expert in plant tissue culture. Some of the listeners may remember back in July, we had a webcast about the topic of micropropagation. Dr. Jones is an expert in that field as well and has much more to contribute. Dylan, will you introduce yourself and tell us a little bit about your company and about your product?
Sure, I'd be happy to. So I'm, as you mentioned, Dylan Wilkes. I'm the CTO of Orange Photonics, and we make a product called Light Lab, which is essentially a liquid chromatograph that's used for testing cannabis products. So this is something that's used for things like potency analysis and some terpene analysis, and we've done a lot of work with using the device in phenotyping in general. So my personal background is in spectroscopy, so this is something I'm using light to measure chemical composition. And whether fortunate or unfortunate, I'm actually a third-generation spectroscopist, so it's something that's been running in our family and to continue the tradition. And so I'm excited to talk on this panel, and hopefully we'll get some good information out there. Thank you.
All right, and Dr. Jones, do you mind introducing yourself and telling us a little bit about your work?
Yeah, my name is Max Jones. I'm an assistant professor in the Department of Plant Agriculture at the University of Guelph. Within that, I'm a part of the Gosling Research Institute for Plant Preservation. And so as the name implies, our main focus is the conservation of endangered species, but we work with a number of horticulture species as well. This includes seed, in vitro collections, and we have a small cryopreservation facility. So cannabis is a logical extension for us, and we recently received a license to work with cannabis on campus. So we're actively engaged in micropropagation and biotechnology research. And my intention is to establish a germplasm collection, a public gene bank, for two purposes. One is the obvious, to conserve and make available cannabis genetics. The other is to support our larger mandate to conserve endangered species. So, you know, we hope to use the economic clout of cannabis to kind of support the broader mission of plant conservation. So that's essentially where we are.
Well, that's a natural lead-in to discussing what our topic is today. Kind of a heady topic that covers a wide range. So just for the benefit of our audience, you know, why do we have someone from a spectroscopy background and someone from a plant preservation background on the same panel? Well, it has to do with what is germplasm. I mean, germplasm is seeds and cuttings, but it's more than that. It's seeds and cuttings that you know something about the genetic potential of, and that includes plant phenotyping. It includes knowing the performance of a plant in various different environments. It adds value when you are able to quickly and hopefully cheaply determine something about a plant phenotype. As an example, just a little story in cannabis. Some experienced cannabis growers may know skunk number 1. It's an old variety that is well known, but in order for it to be made, it took over 9 generations and about 20,000 plants per generation in the last few generations. You had to screen that many plants in order to develop the skunk number 1 variety, which is primarily known for being one that breeds true from seed. So as an example of germplasm, skunk number 1 is good germplasm because it's something that has a known, relatively recorded history, and that's not necessarily the case in other cannabis cultivars. I do want to talk a little bit about the marketplace. Today, we want to talk about kind of what is germplasm and why we want to worry about it, how we make it valuable. And we often use the term in the cannabis industry, genetics. We talk about someone having their hands on some good genetics, things like that. And loosely, often when people use that vernacular term, they really mean germplasm. They don't use that term necessarily, but the term genetics is in the cannabis industry is often usually loosely used to mean germplasm. I want to show a figure here. So the key point of this figure, first of all, it shows corn yields. Along the x-axis is the year from about 1866 to 2017, and then the y-axis is bushels per acre. The main lesson here is that around 1940, corn yields started to increase very significantly and continue to do so until the present day. Now, what happened around 1940? Well, that's when most farmers had finally switched over to using double-cross corn hybrid seed. And it took a while for that to start showing benefits, but right around 1940, the curve starts to go up very, very significantly. When you break this down among all the technological advancements that were happening in agriculture during that period, still about half of that increase is due to the improvement in genetics through breeding alone. One of the reasons that we want to worry about this is because breeding is one of the most effective activities that you can do based on our history with other crop species. Depending on which crop species you're talking about, corn being the most well-studied, about half of the yield gains that we've experienced over the 20th century are due to breeding. And that has to have germplasm as the raw material for that breeding. So I'd like to kind of first have a discussion about regulation. And Dylan, I'd like to throw it to you for a moment, if I may, about how your Light Lab product helps people maintain regulatory compliance. I know you have some products for both hemp and cannabis, terpenes and cannabinoids. Will you just take this opportunity to tell us a little bit more about your product and how people use it?
Sure, I'd be happy to. So basically, if you're looking at the hemp side of things, the regulatory side is very important. So often the regulation requires the THC levels of your plant to be under 0.3%. So in order to do that, you need to, of course, have some genetics that would support that, or some germplasm, I should say, that should support that if we want to be correct. But you also need to be able to test it because not only does the genetics affect it, but also the environmental conditions. So people often use a device like the Light Lab to test their product and make sure that the THC levels are within the legal specification. And if they're not, there are certain actions that we can take, such as harvesting early or possibly destroying the product, that kind of thing. So by testing, they're able to ensure compliance, and ultimately that's going to save them money. On the cannabis side, there's a little bit less on the regulatory side, although the same kind of things are going on. People are looking for strains that have specific characteristics, whether it's a cannabinoid profile or a terpene profile, and it's just sometimes very difficult to send, as I mentioned, the thousands of tests that we require to make stable genetics to a lab. It's quite expensive. So our device is often used in sort of the phenotype and chemotype hunt type situations where you're looking at several different characteristics and want to be able to find out quickly what the results are. So I don't know if that goes over enough, but that's kind of a quick start as to how sort of the testing, chemotyping play into the whole germplasm situation.
No, it's absolutely perfect, because what we're talking about here is germplasm is genetics with value added, and being able to know what the chemotype is value added. I mean, the most obvious case of value that you mentioned is in hemp, being able to test your crop in the field and making sure that the plants that you're growing are under the legal limits. There's some value added right there. We kind of have a really interesting panel here today, because we all live in different regulatory regimes. I'm based in Colorado, and you're based in Massachusetts. Dylan, is that right? You're in Cambridge?
That's actually New Hampshire, but close enough.
New Hampshire. Sorry about that. And Max, you're in Canada, an entirely different country with an entirely different regulatory scheme. Max, do you want to talk a little bit about how the regulatory scheme affect your ambitions to establish a germplasm repository?
Yeah, sure. So I got my research license under the old laws. So cannabis was legalized recreationally in Canada in October. I received my license around the same time, but it's under the old legal system. And in that license, I'm only allowed to collect material from a certain company that I proposed in my license. When I transition into the new system, I'll be able to do research, but I still won't be able to create a gene bank until I basically get regulated under the nursery license. So I need to do jump through a few more hoops before I can actually establish a gene bank. But the regulatory framework is now there that I can do it, and it'll be fully legal at the federal and provincial level. So it's a step forward, but there's a few more hoops I have to go through before I can do that.
Well, that's the problem that everyone faces under any scheme is a brand new regulatory landscape. Research is slow, research takes time, and we're in a gold rush mentality still. Let's see, do we have one thing I really would like to mention is that, this is tricky legal terrain. Make sure you get legal advice. We're not here to offer any kind of legal advice. We're just here to talk about what we would do if we could and how we'd be able to navigate this incredibly harrowing regulatory landscape. Actually, Max, this is time. Will you keep talking and tell us a little bit more about your preservation and propagation work? What have you learned about cannabis so far in terms of preservation or propagation? What are you willing to share with us today?
Yeah, so I'm working with a few different companies, so some of it's a little private, but the one thing I would say is that, there's a lot of our conservation with cannabis at the moment will be clonal, because at the moment, in order to meet the quality assurance regulations in our system anyway, you have to have consistency of product. So if you grow from seed, you have too much variability, so it's all pretty much clonal based. So what this means is that it's all done through either cuttings or micropropagation. I focus mostly on the micropropagation side. And the one thing I have learned is that the protocols that are out there that are published don't seem to be reproducible in our hands. So, I don't doubt that the cultivars they were working with responded how they report, but when you try to apply that to different genetics, it doesn't really work very well. I'm sure you're working in the field as well. You've probably encountered this in your own hands. So there's a lot of work to be done. We're making progress in that direction, but there's still a little bit of ways to go. We've also done some work in cryopreservation, and so we've been able to cryopreserve them and bring them out. So the framework's there. There's still a lot of work to do before it's a refined, reproducible protocol, but it's on its way.
Yeah, I appreciate the need that for you to be discreet since you're working with private companies. So I appreciate anything that you're able to share. My experience too has been that the published protocols are not exactly universal or usable. We had to modify the protocols significantly. And since we're focused on my side of things with trying to figure out how to do this on a mass nursery, mass propagation scale, protocols that were developed for research are not necessarily the same kinds of protocols that you want to use if you're trying to do mass production. And that has to do with labor efficiency, liquid-based versus auger-based and things like that. There are a lot of different tweaks and a lot of different processes that still need to be developed in order to adapt micropropagation to the mass markets, to where you would want to be producing thousands of clones per month instead of just a few dozen, which maybe what you can do with research scale protocols. There's a big gap between.
Yeah, and I'd also mention that if your objective is primarily conservation of genetics, so if you're trying to maintain your genetic library in vitro, your goal is usually to slow the growth down. Whereas if you're trying to use it for large scale micropropagation, your goal would be to speed the growth up, right? So it's two different approaches with two different objectives.
Yeah, you're going in two different directions. Yeah, you're going in two different directions, and you can't rely on the same protocols or even the same thought process when it comes to ironing out the inefficiencies in your process.
Right, and so we're working a little bit in both directions. So, the companies we're working with are interested in both sides of things. They want to preserve their genetics. Some of them are also interested in large scale propagation, but Those are two different things.
Moving on a little bit to phenotyping again. Phenotyping is a measurable property of a plant. So Dylan, what phenotypes are you able to measure now that are commercially ready, that are a part of your platform? Do you mind reviewing those in a little bit of detail for our audience?
Sure, I'd be happy to. So the main thing we focus on is, right now, of course, although we're working on other things, is cannabinoids. So we actually are measuring, I believe we're up to 7 cannabinoids. So just to list them out in a jumble, it's THCA, Delta-9 THC, CBDA, CBD, CBGA, and CBC, and CBN. So those are the 7 that we're currently focused on or can do right off the bat. We also are able to look at terpenes in a semi-quantitative way, and I won't get into a lot of details about why it's semi-quantitative, but we can essentially tell you if you have a low, medium, or high terpene profile in your plant. And often that's enough from a phenotyping perspective to tell whether a plant is worth continuing on if you're doing some kind of a pheno hunt or not. So really what our device, maybe I can give you a quick example of how our device is used. We recently went and did a large-scale pheno hunt with a group called Humboldt Seed Company. And what they essentially did was pick out some physical traits of the plants that they wanted. And they probably narrowed from about 20,000 plants down to, say, a few hundred. And then from those, we ran through several or hundreds of them on Light Labs. And we were able to kind of pick out things that were interesting, things that have a very high terpene content or very interesting THC CBD ratios, that kind of thing. So we even saw some plants that had things like terpene contents of over 10% by weight. So it was an interesting tool to use as part of it. Certainly not the only key to it, but certainly it was a useful tool during that pheno hunt to be able to kind of help differentiate similar plants that have similar physical traits. So.
In that project, were they required to take the plants all the way to flower, or could they test vegetative material with your platform?
You can actually do both. So it depends a little bit on what you're looking for. Of course, a plant that's early on before flowering is going to have significantly lower THC or CBD levels, but they are present, and often we find that the ratio of THC to CBD is relatively stable. It depends a little bit on environmental conditions, but it is relatively stable throughout the life of the plant. So depending on what you're looking for, as soon as you have enough plant material to actually make a run, let's say 0.5 gram or so of material. So it is possible to start really early. In this case, we were looking at flowering plants because most of what they were interested in was the different terpene profiles that were present. And so in that case, the terpene profile really starts to express itself in the flowering mode. So that's where we did that particular test. But we have done earlier testing, and we're actually starting a new project shortly that'll look about 6,000 tests, and it'll be very early level plant testing.
Mitch, we can't hear you there. You may want to unmute or something.
My bad, thanks, Howard.
There we go.
Really, I was curious about that, Dylan. How early in the plant life cycle your platform would allow people to test, if you were limited to flower or if you could use vegetative material?
Yeah, so you definitely can do vegetative material. The answer a little bit is we don't really know, since we haven't done a lot of testing in early plant material, exactly what information may or may not be useful, but it's actually starting the end of this month, we'll have a large-scale test to kind of learn a little bit more about that and look at hundreds of different plants. So we're kind of excited to do that and really understand more about what the plant does sort of early in its life cycle as far as the chemotype is concerned.
I look forward to hearing anything about that you release. If you do a white paper on that, that'd be very, very interesting. There is some indication in the literature that chemotype, at least as far as cannabinoid, chemotype of the vegetative material is predictive of but I don't know about terpenes, and that sounds to me like what you were saying. I understand.
Right, yes. We've seen similar sort of anecdotal, because I don't have enough data to support it from a really quantitative perspective, but certainly when we see plants that express high levels of THC during the vegetative state, it it typically expresses higher levels of THC in the flowering state in the final product. But we haven't seen so far the same correlation with terpene content, but that may be simply because we don't have the data to support it, really. So we're kind of still in a wait and see mode there.
So Max, this kind of leads to a question for you. Do you, in your laboratory, do you work on any plant products, or are you focused solely on propagation and preservation, or do you look at any kind of plant products?
So in our lab, we focus mostly or primarily on in vitro stage and conservation of genetics. I do have some projects going on with producers where we do research in their facilities where we look more at the later stages of production side, so greenhouse production or indoor production and the effects of the environment or nutrient solution on yield as well as chemistry.
I was just curious because I noticed from looking at your research history that you have looked at that in other species, and I was just curious what where you were at on that with this particular species.
Yeah, so just due to regulations at the university, we're limited to doing it in tissue culture because that's what our license is. But moving forward, things will probably change in that regard. But for now, we do that type of research in collaboration with companies.
All right. One missing element in our panel, we weren't able to get someone today to talk about it, but DNA testing is one type of plant phenotyping that can be done at a very early stage because you can do it with tissue culture sized amounts of material. I urge people in the audience to look up Medicinal Genomics if you're not familiar with their work. They just recently released a completed cannabis genome. We weren't able to get someone from that company to be on the panel today, but I definitely wanted to give them a mention because it's a type of testing that once it's figured out can be repeated relatively cheaply. So that's just something for the audience. Another topic to talk about, which is outside of the expertise of our panel, although seed certification. Does anyone in the panel have any experience working with companies that are trying to get seed certified through hemp or otherwise?
I have a couple things to add there. We do have a breeder in Canada that uses our light lab with some of his certification work. He hasn't released any official kind of cultivars, I'd say, that are officially certified, but he has been working with trying to do that. So there is, I know there's work going on. I'm not really particularly involved in that. But the other thing I've seen on the sort of the seed side and certification side is there's a fair amount of work going on, again, through Humboldt Seed and some others that we've worked with that is about stability of the seed strain, or I'm sorry, the seed line. So if you were to plant, 20 seeds from a single batch of a mother plant, then you get a significant variation, and being able to stabilize that is obviously a difficult task, but one we've been working on quite a bit. We look at things like the chemotypes and see how much variation you get in cannabinoid content between different seeds, as well as terpene content, that kind of thing. And it's really interesting when you plant a whole field from seed, how much variation there really is. We did a specific strain that there was about, I want to say 20 plants of, and the variation of even the coloring and the shape of the flower and everything was quite significant still. And this was a relatively stable seed line. So there's still some work to be done there. And I know a lot of people are interested in doing that.
Well, that stabilization work, the skunk #1 example that I introduced earlier, we're talking about 9 generations. That's, at production pace, you can get 4.5 crops a year. But there's a lot of work that goes in there, and it can kind of interfere with production. So when you're a strictly profit-driven or immediate short-term profit-driven organization, it's tricky to do that. So that's going to be the reality for a while until we can do the good work that's necessary to develop these stabilized cultivars, and then we can start talking about crossing them and breeding them and understanding what we're going to get when we do that work.
Absolutely, I agree. And one of the ways that they've been trying, I don't want to say get around it, but a lot of the people we've been discussing with are using strains that have been used for generations in the illegal space or the black market, since they are often used over and over again and tend to stabilize over time, although there's some risks involved with that just from the point of view of not knowing what the genetics really are. So it's going to be a long road for that, but hopefully we'll get there someday.
So Max, I'd like to loop you back in and talk a little bit about this, about your vision for a germplasm repository. Historically, these have been public resources that have been maintained with government approval and government help. How do you see your germplasm repository vision? How do you see that evolving in this regulatory landscape? Do you hope that eventually we'll achieve a status quo that's like what we're used to in croplands, or will we be going someplace entirely different?
Well, my hope is that we'll go somewhere similar to other crops. So if I can get the license to do it, it is fully legal in Canada, so we are capable of doing that. I'm in a public university, so my intention is not, we're not profit driven. My intention is to have a public, non-profit germplasm collection. And I really think it's important to keep it public because private collections, they just don't serve the same purpose. To ensure accessibility, it needs to be public. I would hope long term that the government would decide to fund it long term. In the short term, I envision that I would have to do it through a cost recovery basis. So, like I said, we're not profit driven, but we would need to recoup money to keep the operation going. And as I mentioned in the intro, our real objective here is to do the cost recovery on cannabis to support our larger mandate of plant conservation. So, the cost recovery would cover a lot of our basic operation costs to give us a long-term financial stability for endangered plants. There's a bit of a sidebar for this, but endangered species just don't have the financial resources that the cannabis industry does. So we're hoping that it's a kind of a win-win for both situations.
Well, it's a common problem in conservation. The charismatic megafauna gets the money, and that's the same problem that we're experiencing in cannabis as well.
And I think it's important to collect the genetics now. If you look at the Food and Agriculture Organization of the United Nations estimates we lost 75% of our genetic biodiversity in the 21st century. And basically what happens is as new strains are developed, stable seed lines and stuff like that, they will probably replace a lot of the what will probably be referred to as heirloom varieties one day. So all the varieties that are the cultivars that are out there right now will probably be displaced to a large degree by improved genetics with better disease resistance and higher yields, et cetera, et cetera. But the problem with that is that a lot of the genetic diversity that exists now will be lost, right? So our intention is to collect the genetics now before it's lost, and then have it for future breeding efforts.
Well, there are feral populations all over North America that I'm aware of that need to be collected and cataloged and maintained in some kind of a sensible way so that they can be useful as a reservoir, as a resource for genetic variability.
Right, and I think what's important to note is that from the germplasm collection side of things, it's not all of the elite strains that we're really the most interested in. It's a lot of the feral ones, as you say. They might not be good for production, but they might have resistance to powdery mildew or spider mites or something like that can then be bred into the population of elite varieties. Because I don't think the analysis has really been done, but I think if you did a genetic analysis of the elite cultivars that are available right now, you'd find that it's just a sliver of the diversity that actually exists. And so if we only focus on those, then we would lose a lot of valuable genetic diversity.
That's a very good point. And it's kind of a note that I want to finish on is discussing the other types of traits. We can measure the easy ones. The current obsession with the market is chemotype, which is understandable because that's the marketable aspect of the plant. But you mentioned other things that are just as important to production and important to the bottom line, but they're a little bit indirect. Disease resistance, things that affect the producer's experience, but the consumer may not necessarily be able to see that because of a finished product. Would hide that complexity from them. There are so many other types of phenotyping, which is why I wanted to give medicinal genomics a shout out because of their DNA testing work and their pathogen detection work. Looking for cheap ways to screen those types of phenotypes, which is why I was glad to have Dylan on the panel because they've figured out a good platform, a way to test one important phenotype, but it's only one. Are there other traits? Are there any particular traits of interest? If you could wave a magic wand, Max, what would you want breeders to focus on besides cannabinoids and chemotype?
Well, I really do think disease resistance and insect resistance is number one. For one reason, there's not a lot of pesticides or control options available for producers. And even if there were, consumers don't really want pesticides sprayed on it. So I think it's important to both consumers and producers to focus on disease resistance and insect resistance. And as just to focus back on the genetic markers and stuff, they are very valuable, but I don't think there's any replacement for large-scale traditional breeding programs. Like when you talk about the increase in yields in corn, that was all done through traditional selective breeding by highly qualified plant breeders. I don't think you can replace that with genetic markers. I think it highly complements the process, but you'll never replace it entirely.
You can shave a few generations off with marker-assisted breeding, but you still have to do the breeding. There's no substitute, I agree.
You might increase the efficiency of your breeding program using genetic markers, but they're not in isolation a replacement for traditional breeding.
That's an important point to emphasize, especially for the non-experts in our audience, that the genetic technologies are not going to replace this classical breeding. The classical breeding is the foundation upon which all of that is based. Germplasm is a very heady topic, like I mentioned, but the reason that it's important to the industry is producers are going to need it for the reasons that we've mentioned. Disease resistance. This is going to affect the daily experience of producers who are trying to produce, whether they're hemp growers out in the field or whether they're indoor growers growing under hot light. It's going to be the same kind of need. We need to breed stronger plants that are more resistant, that have the right form, that have a good consistent form that requires less labor. These are all goals that we should have. But one of the reasons that germplasm is an important topic and why the lack of public resources is a real challenge now is a matter of scale. These large-scale breeding programs are exactly what you say. They are large scale, and they may be beyond the reach of the average producer. But with public resources, everyone can perform a little portion of the breeding, and with the proper mechanisms for sharing, people are able then to contribute what they can to breeding programs and result in overall crop improvement. Everybody benefits, and that's the purpose of public resources, is that a little bit of sharing yields benefits to everybody in the industry. That's my soapbox for this topic today. I'd like to offer our panelists a chance for a parting shot. We're just about finished with our time today. Dylan, do you have any remaining thoughts or anything to put a cap on this?
Yeah, I do. Just one more thing, not to beat the dead horse of cannabinoids, but some of the other things that we see when we're talking about genetic diversity and that kind of thing is there's been a fair amount of work that we've been involved with in working with landrace and feral strains. And some of those landrace strains really do have really interesting minor cannabinoids. And some of them have, for example, cannabinoids we haven't even yet identified with our mice. And so it's interesting to see some of those landrace strains sort of at least coming into the scientific community. And I think some of the work that Max is doing is going to be great to be able to preserve some of that, because it's going to be really important in order to make new products that could possibly help more people. So thank you. Of
course, you're welcome. Always welcome to have you on. You're always welcome to be on these panels. How about you, Max? Do you have some parting thoughts?
Well, I guess I would just kind of echo what we were talking about earlier, and I really feel that it's really important to have public support for this, so that it's not only private collections, and so that there's actually a public germplasm bank that anyone can access and benefit from, just like in any other crop.
Well, in progress, we'll be faster, willing to share a little bit.
Right, and so I'm hoping people are willing to generously donate their genetics to gene banks once they're available, and then everyone kind of benefits.
That's the goal, that's the vision. Well, I'd like to thank everybody on our panel today. I don't know if we have any questions from our gracious host. Ellis, do you have any parting shots yourself?
No, I want to just thank everyone for getting on today. This is a big topic of interest in my office particularly, and it's great to get you guys together and hear everyone's perspective and take on the work you're doing in the industry. And so greatly appreciate that. I'm sure our audience does as well. And so, Mitch, appreciate you putting this together and being the host today. And we look forward to our Q&A with our audience. So anything else, Mitch, that you want to say to kind of wrap this up? If not, I can kind of shut this down.
Well, just thank you. I have a question for you, Ellis, in terms of, you said that it's of interest to American cannabis. Can you describe this from a producer standpoint? Because we've got a trio of nerds here that are talking about the things that make their eyes light up. But what is it that makes your eyes light up about this topic from your perspective?
Well, as a cannabis enthusiast for many, many moons, and having a personal experience and passion with it, I've traveled the world and collected genetics from different regions, and those heirlooms or land races are things that I am looking forward to get to put to work and see what information those things can bring benefit to our industry. And so for me, these are the things that we've been strategizing and planning as a company to do this type of research and this type of service offering to the industry with clone businesses and genetic businesses. And so for me, it's been a lifelong work that I've been exploring. And over the last 4 years, I've been working with you, Mitch, to really bring this to light on what we've been talking about today in some form or fashion. And so I'm excited to see kind of where this industry is going and really pull, a lot of these varieties from around the world, but also these local ones that we've seen from Kansas to Michigan to Wisconsin to all these areas as well, that is feral seed that have a lot of codes that need to be unlocked for us. So I'm excited to see where the future holds with our industry as rules and regulations lighten up, give us more freedom.
Well, thanks for chiming in. I wanted to hear your input as well, because germplasm is a heavy, wide-ranging topic that can be approached from many, many perspectives, and it needs to be approached from many perspectives because it's so complex. So thanks for your input. The only parting shot that I have is a hearty thank you to our panel for being with us today, and a thank you to our audience for listening. I'd like to open it up for questions.
Great. Well, we'll wrap this up. We look forward to the Q&A here with our audience. I'd like to thank everyone on the panel today, Dylan Wilkes, Max Jones, Dr. Mitch Day. Also like to thank our sponsor, Cannabis Tech. I'm Ellis Smith, and we look forward to our Q&A. Thank you very much for your time.